Yes, we are cruising steadily in the India's biggest Med PG Prep visual extravaganza, the Dempsey Medicos DVT 2023. It is my pleasure to introduce our next faculty. And let me also tell you, the next session is Biochemistry. We are honored to have with us Dr. Sonam Bhatia, who will be taking up the topic of biochemistry and share her insights and experiences with you. Dr. Sonam Bhatia has done her MBBS from GMC Patiala. She did her MD from Maulana Azad Medical College, New Delhi, and she also did DNB Biochemistry. Well, without any further ado, let's welcome our esteemed faculty for Dempsey Medicos, the very learned Dr. Sonam Bhatia, who will be taking the topic of biochemistry. Over to you, ma'am.
Hello everyone, excited for biochemistry? Right, join back. Very good afternoon. So we are here to discuss last minute revision topics, right? And I'll be covering vitamins along with some questions on whatever is important for minerals, electron transport chain. We will be discussing some important topics on molecular biology as well. Right, only the most important topics. Do not worry. I know you all have already gone through all this. You already know this, probably even now better than me. So let's continue and let's start with fat soluble vitamins. So we are starting with biochemistry. We are starting with vitamins. We know that there are two types of vitamins, fat-soluble vitamins and water-soluble vitamins. The fat-soluble vitamins are 4, A, D, E and K, and water-soluble vitamins are mainly the B complex, which itself has many other vitamins, and we have vitamin C, ascorbic acid. Out of the two, we require chylomicron for the absorption of fat-soluble vitamins. The water-soluble vitamins do not require the chylomicron. They will be absorbed as such, right? VLDL, however, is the lipoprotein with the maximum concentration for the fat-soluble vitamins. We do not have, VLDL does not have water-soluble vitamins. We do not need actually lipoproteins for the water-soluble vitamins. We only need lipoproteins for the transport of fat-soluble vitamins. And out of the two, the chylomicron is for the absorption and VLDL is one which is carrying the maximum concentration of fat-soluble vitamins. Can they be stored? Yes. Out of the two, fat-soluble vitamins can be stored, and we have the water-soluble vitamins which cannot be stored. And where are they stored? The two tissues are liver and adipose tissue. Now, one more thing. Although water-soluble vitamins, they cannot be stored, but some water-soluble vitamins like you have vitamin B12 and vitamin B6. They can be stored in liver. Right. Alright. Now, out of the two, fat-soluble vitamins and water-soluble vitamins, which one is excreted in urine? Yes, you know this. Fat-soluble vitamins are not excreted. It is water-soluble vitamin which will be excreted in urine, right? So these are some major points about the two fat-soluble and water-soluble vitamins, right? Alright. Now, one more thing. Vitamin A is an isoprene derivative. We are first starting with the fat-soluble vitamins and we are starting with the vitamin A, then D, then E and then finally K. So vitamin A is an isoprene derivative. It is a long structure And you do not have to remember the structure. Just remember that vitamin A is an isoprene derivative. 6 microgram of vitamin of beta carotene is responsible to give you 1 microgram of active form of vitamin A. Now, if somebody asks us what are the active forms of vitamin A, we have 3 of them. Number 1, of course, is retinal. Then we have retinol, and then we have retinoic acid. And can I choose between them that which one is the maximum, which one is most active? No, there is nothing like most active. There is nothing like that which one of these is inactive form. No, all of these 3 are your active form. Retinal, which is for retinaldehyde, retinol, which is the alcoholic form of retinal, and retinoic acid, which is the acidic form of retinol, right? Then the functions of vitamin A. The phototransduction cycle. It is very famous that vitamin A deficiency is responsible for night blindness. Rather, it is the most common cause of night blindness worldwide, right? Now, how is vitamin A responsible for the light in the dim vision, or it is responsible for dim vision? So this is through a signaling pathway of rhodopsin and rod cells. Let's see, this here is the rod cell. The rod cell gets a signal which is already having disc-like structures. The signal is in the form of light. When the light of a particular wavelength falls on the rod cells, there occurs the transformational changes. In this transformational changes, there are 2 things. Please note, when the light falls on the rod cells, there occurs transformational changes, conformational changes, and there are 2 things in this conformational change. Number 1 is of course the pigment rhodopsin, and another thing, number 2, is vitamin A. So vitamin A will have a very small but a very significant role in phototransduction cycle. Vitamin A and rhodopsin, they will combine only when there is a dim light falling on the rhodopsin. When this happens, there is a protein known as transducin. Write this down. Transducin. There is a protein known as transducin. This transducin undergoes a conformational change, and due to this conformational change, it was earlier GDP bind, but now instead of GDP you can see that it binds to GTP. And once this is active, there is another activation complex known as phosphodiesterase complex which gets active. Once phosphodiesterase complex is active, it converts cyclic GMP to 5' GMP. It converts cyclic GMP to 5' GMP. Now see, one important thing here. There are 2-3 key important points in this diagram. Point number 1, what is this transducin doing? This transducin was earlier GDP bound and it now binds to GTP and is active. What it does? Point number 2, it activates phosphodiesterase. And what does phosphodiesterase do? It converts cyclic GMP to 5' GMP. Now what is cyclic GMP responsible for? The cyclic GMP is responsible for keeping the sodium ion channels open. But when the transducin gets activated, phosphodiesterase complex get activated. As a result, what will happen is, now this will get closed. The sodium ion channels are open when there is cyclic GMP, but because cyclic GMP got converted to 5' GMP, now these channels, they have closed. Now these channels, they have closed, right? And that is where once the closure has taken place, all the sodium ion concentration will be high on one side, on the outer side of the membrane, which is a signal to the next bipolar neuron. From there, the signal will go to the optic nerve and finally to the brain ophthalmic region where the actual vision will be formed. But what will be the question on this? Transducin and the role of transducin. Very important, and the role of cyclic GMP. Two things. Transducin, what is transducin doing? It is causing phosphodiesterase activation. Point number 2, what is cyclic GMP doing? It is responsible for opening of sodium ion channels. Then, besides this, which form of vitamin A is responsible for growth and differentiation of the cells? The answer is retinoic acid. It is retinoic acid which is responsible for growth and differentiation. For the treatment, we use vitamin A in AML cases, for measles, for acne, xeroderma pigmentosa. In both of them, last one, we are using topical ointment in the form of isotretinoin. These are the functions of vitamin A. Also, vitamin A is an antioxidant along with vitamin C and E. Which is the most potent antioxidant? Quickly tell me in the chat box. The deficiency. Most common cause of preventable blindness worldwide. The first symptom will be nyctalopia, night blindness. The first sign will be the conjunctival dryness. If this continues, it will result in corneal dryness. Dryness of cornea can ultimately result in keratomalacia, that is liquefaction of cornea. However, the confirmatory sign is Bitot spot. Deficiency of vitamin A is also associated with poor wound healing, probably because it is responsible for growth and differentiation of the cells. But if you see this term poor wound healing, there are two vitamins which should come to your mind, that is vitamin C and vitamin A. You will mark vitamin A only if vitamin C is not given in the options. So yes, it is C more than A. Deficiency of vitamin A is responsible for recurrent respiratory tract infections, and that is why it was also given as supplementation during COVID-19 infections. Then hypervitaminosis A. It causes headache, anorexia, alopecia, bone pain, dry skin, drowsiness. Most important feature of all of these is headache. It is a pseudo-tumor cerebri, raised intracranial pressure that occurs because of hypervitaminosis A. It will mimic tumor-like headache, but when the patient is asked to get an MRI done, there will be no space-occupying lesion. Vitamin A is contraindicated during pregnancy because it can result in injury to lysosomes. So we have now completed vitamin A. Let's see vitamin D. Vitamin D. Vitamin D3, what is the most important action is that it looks like steroid hormone. It has a function like steroid hormone. It mimics the action of steroid hormone. Yes, vitamin D3 can be formed by us. We need sunlight for it, but do not put sunlight as a source for vitamin D3. When the sources are there, we will put our two sources. One is halibut liver oil and cod liver oil. These are rich sources for both vitamin A as well as vitamin D. Vitamin A as well as vitamin D. Right? And then we have 7-dehydrocholesterol, which in sunlight will be converted to cholecalciferol. Now this cholecalciferol is converted in the liver to 25 hydroxy vitamin D, and this form of vitamin D is formed by the enzyme 25 alpha hydroxylase, which is present in liver. And then we have an enzyme known as 1 alpha hydroxylase, which is present in the kidneys, and this forms calcitriol. This forms calcitriol, vitamin D3. So the active form of vitamin D3 is 125 hydroxy vitamin D, right? We call it calcitriol, right? And you will find that this term is being interchangeably used for calcitriol and 125 hydroxy vitamin D3 or D3 simply, all of them is same. The function of vitamin D3 is that it is responsible for calcium homeostasis. It acts on 3 tissues: intestine, kidneys and bones. At the level of intestine, it is causing absorption of calcium. At the levels of kidney, it causes reabsorption of calcium. And at the level of bones, it is responsible for bone remodeling. In all these 3 tissues, the receptors are different for calcium absorption, for reabsorption and for bone remodeling. We have TRPV6 receptor at the level of intestine, we have TRPV5 receptor at the level of kidneys, and rank ligand at the level of bones for bone remodeling. Then vitamin E, tocopherol. It is also an isoprene derivative. When we were discussing vitamin A, we said that is also an isoprene derivative. This one is also isoprene derivative. Now please note, A and D, the rich source was fish oil. For vitamin E, it is vegetable oil which is the rich sources. Wheat germ oil, rice bran oil, sunflower oil, safflower oil, all of these are the rich sources of vitamin E. They are also rich in essential fatty acids, and hence the deficiency of both essential fatty acids and vitamin E will occur simultaneously. But it is one such vitamin which is having least deficiency. Of all the other vitamins, deficiency of vitamin E is least. Anyhow, water soluble vitamin deficiencies is much more common than fat soluble vitamins. These, because they can be stored in the liver in adipose tissue, that is why they produce toxicity, and that is the reason they are having very high, you will usually never see a clinical case for vitamin E deficiency or your vitamin A, D, E, K deficiency, usually never seen. So vitamin E is found in the vegetable oils. Fish oil rich in A and D. Vitamin E is most potent antioxidant, and yes, it prevents the biological membranes. Most of the students have answered it correctly, and that is vitamin E is the most potent antioxidant. What it does is it prevents the peroxidation of the lipid membranes, because the membranes are rich in phospholipids. So the membranes, if they will get peroxidized, and if vitamin E is there, it will take away that free radical from the membrane, and because vitamin E sooner or later will go out of the cell, it will take that free radical along with it and leave the cell. The damaging to the lipid membrane will be least if vitamin E is present. And yes, it is the most potent antioxidant. The next vitamin, from vitamin K to all the other vitamins, these are the most important vitamins, alright? So starting with vitamin K, there are 3 forms of vitamin K, K1, K2 and K3. We will get K1 from plants and we call it phyloquinone. We get K2 from animals and we call it menaquinone. We get K3, which is synthetically made, and it has been made to be water soluble. It has been made to be water soluble. And this one, with the one which has been made to be water soluble, it will be it has been made synthetically. And hence you will see that most of the time we write vitamin K as hydroxyquinone. It is the active form of vitamin K. So whenever you hear these terms, phylloquinone, menaquinone, menadione, we are having quinone forms. These are all the forms of vitamin K, but the active form will be your hydroxyquinone or simply hydroquinone. Now, what does vitamin K do? What is the function of vitamin K? Very important slide. Do not ask me to fasten up. Do not worry. I will be freeing you in 1 hour, right? So just take this as an opportunity to revise with me, because I know you're not going to revise this a lot. So just take it as an opportunity to revise. So let's discuss the function of vitamin K. The post-translational modification of factor 2, 7, 9, 10 along with protein C, protein S, osteocalcin, osteonectin, all these proteins are activated by the vitamin K. The vitamin K in its hydroxyquinone form gets converted to its epoxide form. Now, during this conversion from hydroxyquinone or hydroquinone to epoxide conversion, please note the change that is occurring in the factors. The factors are protein. Notice the pink colored protein, it gets green colored, right? And this requires a carboxylase, and this carboxylase requires vitamin K. Simply, if you see the change, what is the change? The change is occurring at the level of glutamic acid. The glutamic acid was earlier having only one COO negative. On gamma carboxylation, it gets 2 COO negative, and the 2 COO negative can chelate calcium because calcium is Ca2 positive. And this gamma glutamic acid is known as GLA. It is carboxylated glutamic acid and is the active form of vitamin K. Now, during this reaction, we got an inactive form of vitamin K and we need to convert it to the active form. For this, we need an epoxide reductase, and this epoxide reductase is inhibited by a group of drugs known as warfarin. These warfarin and dicumarol, these drugs, they inhibit this and hence they are used as anticoagulants. Now, what is deficiency of vitamin K associated with? The only finding will be prolonged PT, prothrombin time. And the deficiency will result in hemorrhagic disease of newborn. And hence newborns are supplemented with two things. We give them vitamin K and vitamin D. These are the two things which we give. Alright, now we are starting with the water-soluble vitamins. The water-soluble vitamins, we are starting with vitamin C. We have L-gulonolactone oxidase absent in humans, and hence vitamin C is essential for us. Otherwise, it would have been synthesized by us. Then the milk is deficient in two things. Remember, the two things which milk is deficient in, that is vitamin C and iron, and those two things are required by this enzyme prolyl hydroxylase, vitamin C and iron. Now, why do we need this enzyme prolyl hydroxylase? Because it is responsible for a post-translational modification. It causes post-translational modification of proline. Please note, similar modification we have seen for vitamin K, but that was a carboxylation. If the question says post-translational modification and carboxylation, the answer will be vitamin K. But if the question says it's a hydroxylation which is due to the, it is a hydroxylation post-translational modification, then the answer will be vitamin C. Now, this prolyl hydroxylase is converting proline to hydroxyproline. Structure is not at all important. The only thing you have to remember is that hydroxyproline is unique to collagen. There is no other vitamin which has, there is no other protein which has the hydroxyproline. Hydroxyproline is an amino acid, a modified amino acid that exists only in the protein collagen. No other protein has it. And what is collagen made of? It is made of repetition of glycine XY. In 33% of collagen is glycine because every third position will be occupied by glycine. And then you have somewhere proline. Most of the spaces are proline and lysine. Now, another important thing is a similar modification by an enzyme lysyl hydroxylase occurs for lysine. It is hydroxylation of lysine, but that is never asked. They are only and only asking proline hydroxylase. Do not worry, lysyl hydroxylase will not be asked, but proline hydroxylase is very important. Then, vitamin C deficiency results in bleeding of gums, poor wound healing. Because it is resulting in bleeding, it can present as anemia, corkscrew-like appearance of hairs. Most of these symptoms can be explained by the hydroxylation of collagen. Besides this is vitamin C is also responsible for many other hydroxylations. Like we have with dopamine beta hydroxylase, that hydroxylation also requires vitamin C. There is a selvin alpha hydroxylase. It is a rate limiting enzyme for bile acid synthesis that also requires vitamin C. Hydroxylation which is occurring in the adrenal cortex for active steroidogenesis, that also requires vitamin C. So most of the hydroxylation reactions do require vitamin C. So key term, if you see carboxylation, the answer is vitamin K. If you see hydroxylation, the answer is vitamin C. Is it that all the hydroxylation requires vitamin C? No. Only these are some few major examples which you have to remember. In addition, vitamin C is required for the absorption of iron. We absorb dietary iron with the help of a transporter known as DMT1. But whatever iron we eat in diet is in the form of ferric iron. It is converted to ferrous with the help of vitamin C. It is converted to ferrous with the help of vitamin C. So, vitamin C is responsible for the conversion of ferric to ferrous and then the absorption of the iron. And that is why pregnancy, we advise the pregnant female to take iron along with vitamin C. Right? Then, vitamin B complex. Vitamin B complex. So, we are starting now with vitamin B complex. Vitamins is very important. Vitamins, and whenever you are revising, do not rush into vitamins, especially now. Those who, most of us are going to give the examination now, and this was, most of the questions were in biochemistry were from vitamins. So do not rush when you are revising vitamins. Then we come to vitamin B complex. We are starting with B1, thymine. But the thymine is required in the form of thymine diphosphate. Please note, there are two forms of thymine. One is diphosphate and another is triphosphate. The diphosphate is required for two reactions. Number 1, oxidative decarboxylation. The oxidative decarboxylation reactions are 3 in number. We have number 1, PDH, for pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, and the third one is branch chain ketoacid dehydrogenase. Quickly tell me in the chat box, if this enzyme branch chain ketoacid dehydrogenase is deficient, which disorder will it be resulting in? Meanwhile, I'll discuss B1 and I'll take the answer in the end. So we have thymine diphosphate, which is required for number 1 oxidative decarboxylation. There is one more thing. The oxidative decarboxylation not only requires thymine, but it requires 5 coenzymes. Those 5 coenzymes are thymine, lipoic acid, coenzyme A, FAD, and NAD. Thymine is B1, lipoic acid is not a vitamin, coenzyme A is the active form of vitamin B5, FAD is the active form of vitamin B2, NAD is the active form of vitamin B3. So these 5 factors are required for these 3 enzymes. All of them require these 5. They have a similar mechanism of action. Another reaction is transketolase. Very important. This was last year's NEET question. Transketolase, erythrocyte transketolase is used to assess vitamin B1 status. Another question from thymine NEET was Wernicke-Korsakoff psychosis, global confusion, ophthalmoplegia, and ataxia. These are the symptoms which are seen due to vitamin B1 deficiency. Now, the reason for these symptoms is the second form of thymine. Thymine is also required as thymine triphosphate for the nerve and muscle conduction. And that is why B1 deficiency not only causes Wernicke-Korsakoff psychosis in alcoholics, but is also responsible for dry beriberi, wet beriberi, and Soshin beriberi and infantile beriberi. Now please try and understand. If you see a case of thymine deficiency, what will be the symptoms with which the patient will present? If it is dry beriberi, it will present with peripheral neuropathy. If it is wet beriberi, then there will be symptom of dry as well as there will be edema seen, right? And that is due to associated congestive heart failure which has resulted into it. And then we have the Soshin beriberi, which is acute fulminant wet beriberi. Now, there is the Soshin is nothing but the wet beriberi gone wrong and it is occurring all suddenly. Infantile beriberi is a type of beriberi which is seen in infants whose mother is deficient in B1. Of course, this infant is exclusively breastfed and that is the reason it is mother's deficiency which is presenting in the infant, right? So thymine, 2 questions, very important. Wernicke-Korsakoff psychosis and transketolase. This transketolase activity is used to assess vitamin B1 status and global confusion, ophthalmoplegia, ataxia seen in alcoholics. In alcoholics, few vitamin deficiencies are more common. And these are B1, B6, B9, B12, B2, B3. They can also happen. See, any water-soluble vitamin deficiency can occur. But the most common ones are due to the B1 deficiency. Most common one. is due to B1 deficiency in alcoholics, and that is seen as Wernicke-Korsakoff psychosis. Now, the enzyme PDH and alpha-ketoglutarate dehydrogenase, these two enzymes are responsible for the energy production. They are part of glycolysis, aerobic glycolysis, and the complete aerobic glycolysis includes TCA cycle. Another important point, if you see a question where they are talking about energy production is decreased, or there is low energy production, or they have presented a female who is having a low energy, who does not feel like going to work or does not feel like doing anything at home, because she is saying that it is low energy, that energy can be due to the PDH and alpha-ketoglutarate dehydrogenase being involved, and hence mark vitamin B1 for it. B1 is for that. Then another important thing. As far as B1 is concerned, deficiency of branch chain keto acid dehydrogenase, and I guess most of you have written it correctly. Yes, it is maple syrup urine disease. The next is B2. B2 will be required for anywhere we have seen FAD or FMN. Usually these reactions are not asked, but the most important one is erythrocyte glutathione reductase. This enzyme is used to estimate B2 status. And like B1 was asked last year, please note B2 can be asked this year. So it is very important. Erythrocyte glutathione reductase is used to assess vitamin B2 status of an individual. What will its deficiency result in? Non-specific signs and symptoms. Chelosis, angular stomatitis, glossitis, seborrhea, photophobia, dry skin, all these. The next is niacin. Niacin is required for any reaction which requires NAD, NADP, nicotinic acid, adenine dinucleotide or phosphate attached to it. Now, the deficiency of niacin is associated with 4 Ds: diarrhea, dermatitis, dementia, and death. So, these are pellagra. Now, not only niacin deficiency, but also B6 deficiency and also tryptophan deficiency can result in pellagra-like signs and symptoms. So it is 60 micrograms of tryptophan forms only 1 microgram of niacin, but is very essential. Hence, any cause which results in tryptophan deficiency can also result in niacin deficiency. Then we have staple food like sorghum, maize. They are deficient in tryptophan, hence they can result in pellagra. Heart naps is a tryptophan transporter defect. It causes decrease in the reabsorption of tryptophan, hence it can result in B6 deficiency. Per se, B6 deficiency can result in this pellagra-like signs and symptoms, and carcinoid syndrome where there is serotonin excess, because all the tryptophan will be diverted to form your serotonin. There will be no tryptophan left to form niacin. Hence, all of these are causes of aniacinosis. Aniacinosis is nothing but pellagra. The next is B5. Usually, another least common deficiency of all the water soluble is B5. You'll almost never see a clinical case of B5 deficiency, and that is burning feet syndrome. Where is B5 required? Anywhere we write coenzyme A, acetylation reactions, acetyl-CoA, TCA requires acetyl-CoA. Lipid synthesis requires acetyl-CoA. Any reactions where we require acetyl-CoA will require B5. B6, pyridoxal phosphate. Mark this, another very important vitamin. Pyridoxal phosphate is the most important vitamin for protein metabolism because it is required for transamination reactions. What are the transamination reaction? Put it in the chat box. I'll see it in the end of vitamin B6. The transsulfuration pathway also requires, transsulfuration means conversion of methionine to cysteine. There is an enzyme cystathionine beta synthase that also requires vitamin B6. Then there is decarboxylation reactions. They also require vitamin B6. Glycogen phosphorylase that also requires vitamin B6. Hydroxymethyl transferase to convert glycine to serine that also requires vitamin B6. This one, kynureninase, very important. Kynureninase tryptophan metabolism is an enzyme. If B6 is deficient, then kynureninase will not function. Kynurenin will accumulate and result in formation of xanthurenic acid. And this is seen in tryptophan load test. It is a test to assess whether a person is having adequate quantity of B6 or not. Then we use vitamin B6 for heme synthesis, ALA synthase. The first rate limiting enzyme for heme requires vitamin B6. Deficiency of vitamin B6 can result in peripheral neuropathy. It can result in sideroblastic anemia because heme synthase, the enzyme ALA synthase requires it. Even vitamin B6 deficiency can result in xanthurenic aciduria. But this only happens if we give large quantities of tryptophan. And vitamin B6 is the most important vitamin for protein metabolism. One key and most important point is transamination reactions. And what are transamination reactions? Yes, ALT, AST. We no more call it SGOT, SGPT. The new terminology is ALT, AST. And what is Hartnupp's disease? Some of us are asking questions in the chat box. The Hartnupp's disease is a renal tubular transporter defect, as a result of which there will be loss of tryptophan in urine. Now, because tryptophan is not reabsorbed from the urine, there can be deficiency of vitamin B3. So, in Hartnupp's disease, there can be pellagra-like signs and symptoms. Focus on pellagra, any cause of pellagra. And the classical 4Ds for pellagra is, yes, diarrhea, then Castle's necklace, dermatitis. It affects C3, C4 dermatomes causing classical Castle's necklace. Then there will be dementia and death. Now coming to B7. B7, biotin is deficiency is rare but can occur in people who are taking Avidin. Avidin is supposedly present in raw eggs. So people who consume lot of raw eggs, they tend to develop deficiency of vitamin H or biotin. Now, by and large, vitamin H deficiency is extremely rare because there are few vitamins which are synthesized by our intestinal bacteria. Name them in the chat box. I have showed this during the vitamin K. So one vitamin you can guess is vitamin K. Then there are other vitamins also which are synthesized by intestinal bacteria. Quickly name all the vitamins synthesized by the intestinal bacteria in the chat box. Except vitamin K and H. These two only I have told you. Let's see how many of you have paid attention to that slide. Then deficiency can result in alopecia and dermatitis, and that is why you will find that there are many biotin-related shampoos which are available. The basic funda behind that is that biotin deficiency results in alopecia. Then there are carboxylases. The 3 carboxylases which are most important for you to remember as far as biotin is concerned is pyruvate carboxylase. Please note, pyruvate carboxylase is also known as anaplerotic reaction. Anaplerotic reaction. Pyruvate carboxylase, propionyl-CoA carboxylase and acetyl-CoA carboxylase. These are the 3 carboxylases which requires vitamin B7 biotin. Then the next is B9, folic acid. We have B9. Now B9, folic acid, is responsible for 1-carbon metabolism. The form of folate which we eat is converted to dihydrofolate. This is DHF, dihydrofolate, which is converted to tetrahydrofolate. It is this tetrahydrofolate which is converted to N5N10 methylene tetrahydrofolate. This is my central folate compound. Now, as you can see in this diagram, it can be converted to so many other forms and is required for so many other things. But the major one is conversion of uracil to thymine. Uracil to thymine. So, uracil becomes thymine, or the formation of thymine is where you require N5N10 methylene tetrahydrofolate. N5N10 methylene tetrahydrofolate. Now this N5N10 methylene tetrahydrofolate converts uracil to thymine. It forms serine, converts glycine to serine, and it does many other functions. But the most important one is conversion of uracil to thymine. But this is not one-step process. The conversion of uracil to thymine is not one-step process. You require here in addition vitamin B12, and hence B9 and B12 deficiency both can present with megaloblastic anemia. Both can present with megaloblastic anemia. So B12, our next vitamin is cyanocobalamin. It is having a cobalt ring and it is having a corin ring which is cobalt containing compound. And now vitamin B12 is absorbed along with an intrinsic factor, and hence deficiency of intrinsic factor can also present with megaloblastic anemia. And deficiency of intrinsic factor is known as pernicious anemia. But it will present same as that of megaloblastic anemia. Only the cause of B12 deficiency here is intrinsic factor. Then absorption from the terminal ileum takes place for vitamin B12, and hence people who are strict vegans, because there is no vegetarian source for vitamin B12, or who are having terminal ileal resectomy, these are the people who can develop B12 deficiency. Two most important reactions which require vitamin B12. One is homocysteine methyltransferase and another one is methyl malonyl-CoA mutase. These are the two most important ones. B12, these are the gold. See, the gold standard for the diagnosis for megaloblastic anemia will always be B9 and B12 levels. But in the examination, they usually ask that what will happen to methyl malonyl-CoA levels if B9 and B12 is deficient. If B9 is deficient, methyl malonyl-CoA is normal. But if B12 is deficient, methyl malonyl-CoA will be high because of this enzyme. This enzyme requires the vitamin B12, and The another problem with methyl malonyl CoA is it interferes with the normal sheath formation, and hence myelin degradation takes place, resulting in subacute combined degeneration. Just remember the two enzymes. This one, leucine aminomutase, you may or may not remember, but these two are very important. And when we were noting this down, the conversion of uracil to thymine, we said that B12 is required here. It is through this enzyme, homocysteine methyltransferase. The homocysteine methyltransferase converts homocysteine to methionine. It is this methionine which converts uracil to thymine. And hence both B9 and B12 are responsible for megaloblastic anemia if these two vitamins are deficient. Then these are the antioxidants. Yes, we have discussed that vitamin A, C and E are the primary antioxidants. Then we have polyphenols, ethyl acetate. Then there are certain indirect antioxidants. Out of them, the most important antioxidants are superoxide dismutase, glutathione peroxidase. As far as examination is concerned, most important questions are on vitamin. If on antioxidant, the most potent will be vitamin E. Then this is the whole list of all the antioxidants, but the most important enzymatic is superoxide dismutase, glutathione peroxidase, right? Then coming to the alpha helix and beta pleated sheets, some questions here. Of all the protein structures, the most important question is always on alpha helix. Please note the helix breaker. There are two helix breakers as far as the amino acids are concerned, proline and glycine. You will mark glycine only if proline is not given. Helix is a helical structure. It is a polypeptide helical structure, and it can exist as right-handed and left-handed helix, but right-handed helix is much more stable. Then there are two amino acids which are helix breaker, of which proline is the most important one. Then coming to electron transport chain. I'll just give you a brief of electron transport chain. These are large complexes which are present in inner mitochondrial membrane. The electron transport chain gets its electron from NADH. It will pass on that electron to ubiquinone. There are two mobile segments. You have ubiquinone as mobile segments and cytochrome small c as mobile segments. These are the two mobile segments. Now the electrons are transported from the matrix to the intermembrane space, but for this transport of electron to occur, simultaneously there will be crossing of protons as well. So what electron transport chain is doing is, it is transporting electron from one complex to the another. Simultaneously, protons are getting crossed across the membrane. This is coupled to ATP synthase, and this will bring the protons inside. The key important points. Who is the final electron acceptor? The answer is oxygen. Which is the most irreversible complex in this? The answer is complex 4. Which is the only copper containing complex? The answer is complex 4. They all have iron, but the copper containing complex is only complex 4. Where does ATP synthesis takes place? In FOF1 subunit. FO is present embedded in the membrane, F1 is towards the matrix side. Who are the mobile carriers of electron transport chain? You have two, ubiquinone and cytochrome small c. All others are fixed in the inner mitochondrial membrane except complex 2. It is towards the matrix side and does not transport protons. Does not transport protons. Who is the final decider for whether the electron transport chain will occur or not? The whole control is on one molecule. The controller of respiration is ADP. I repeat, key important points. The final electron acceptor is oxygen. Copper containing complex, complex 4. The copper containing complex, complex 4, the irreversible, the most irreversible reaction is complex 4. Another name for complex 4 is cytochrome oxidase. Where does ATP synthesis takes place? FO-F1 complex. FO is in the membrane, F1 is towards the matrix side. Which compound is the mobile segment of electron transport chain? Ubiquinone and cytochrome small c. And who is the ultimate controller of the whole electron transport chain? That is ADP. Now, coming to the electron transport chain, the inhibitors. For complex 1, amobarbital, it's a barbiturate, retinone, pyridine A. For complex 2, this is important, malonate. Malonate inhibits an enzyme, succinate dehydrogenase. It inhibits the enzyme succinate dehydrogenase. Antimycin A and British antileucide inhibits complex 3. Carbon monoxide, cyanide, hydrogen sulfide, azide, complex 4. Another most important one. Oligomycin is for FO-F1. The O is for oligomycin. The inhibitor to ATP-ADP translocase are two. One is boncreic acid and another one is atractyloside. So we have two inhibitors, atractyloside and boncreic acid. The uncouplers are 2,4-dinitrophenol, 2,4-dinitrocresol and valinomycin. These are synthetic uncouplers. All of them are synthetic uncouplers. And we have one natural uncoupler, that is thermogenin. There are others also, but this is the only one which is asked, thermogenin. UCP1 protein, uncoupler protein 1. This is present in brown adipose tissue. Brown adipose tissue is present in nape of neck region of the newborns and it prevents them from hypothermia. Remember, thermogenin is present in brown adipose tissue and is a natural uncoupler. And finally, we come to our last section, and that is genetics. We are starting with DNA replication. The key important points for this from this slide. Yes, you have to know everything about DNA polymerases. Another important thing. There is something known as Okazaki fragments. Tell me whether these are formed on the leading strand or they are formed on the lagging strand. I'll see it towards the end. Then, leading or lagging, Okazaki fragments. The leading strand is where you are having DNA polymerase attached and RNA primer attached. The lagging strand will also have the DNA polymerase attached as well as RNA primer attached. Now the important points from this slide are two, that DNA polymerase and where is Okazaki fragments formed. And yes, you all know it, that Okazaki fragments are formed on the lagging strand. Now, what are the proteins involved in DNA replication? Their only questions are from the proteins, and then we will discuss DNA polymerase. Number one, of course, is helicase. It will unwind the DNA. Number two, single strand binding proteins. It will cause binding of the single strand on the DNA so that the two do not reanneal. Number three, we have a primase activity for the formation of RNA primers. Please note, during DNA replication, we are using RNA primer. There is a process known as PCR, polymerase chain reaction. There we use directly DNA primers. There we use directly DNA primers. Then we have DNA polymerase for polymerization of DNA. We have topoisomerase. Whatever topological change is occurring during the DNA formation, to unwind that we have topoisomerase. And then we have DNA ligase to join your Okazaki fragments which are formed on lagging strand. Then coming to the function of DNA polymerase. Now very important, most important of all these DNA polymerase is DNA polymerase 1. There are 3 DNA polymerases in prokaryotes, 1, 2 and 3. There are 5 DNA polymerase in eukaryotes, beta, gamma, epsilon, alpha, delta. The function of each DNA polymerase is mentioned in front of each of them, right? But the most important one of all these is the one which is most frequently asked is DNA polymerase 1. The DNA polymerase 1 is having 3 functions. There is a larger segment or the larger fragment known as lino fragment. Lino, L for large fragment. K is silent. So this large fragment is having 2 activities. One is polymerization activity and another is 3 to 5 prime exonuclease activity. Please note that 5 to 3 exonuclease activity is not included in the DNA polymerase 1 lino fragment. That is the only activity which it does not have. So the DNA polymerase 1, the lino fragment, is having 2 activities. One is DNA polymerase and another is 3 to 5 exonuclease. The next enzyme is HTRT, human telomerase reverse transcriptase. Please note it is a reverse transcriptase. Like HIV virus is having a reverse transcriptase, this enzyme will also synthesize RNA to DNA. That means it is converting RNA to DNA. So it is RNA dependent DNA polymerization. Note this down. DNA polymerization. And then we have, what does it replicate? It is responsible for replication of only telomere segment. Only for TNA or only for telomerase replication. Then the activity of the human telomerase reverse transcriptase is absent in somatic cells and hence it results in aging. It results in aging. The germ cell or stem cell, there the activity is present. However, the activity is increased in cancer cells. The activity is increased in cancer cells, and that is the reason cancer cells never age, they never die, they are immortal. Now, as you can see here in this diagram, the human telomerase reverse transcriptase, it is itself an enzyme which is having a RNA segment in it. This RNA synthesizes DNA on the 3' overhang. It will elongate this segment so that the DNA polymerase can extend this lower portion. The mechanism in detail is not important. Key points from this diagram: what is the telomerase adding? It is adding TTA triple G hexanucleotide repeat. So I repeat, what is telomerase adding? It is adding TTA triple G hexanucleotide repeats. These are 6 letters that keep on repeating. What is telomerase doing? It is an RNA-dependent DNA polymerization. It is synthesizing DNA over an RNA. This blue segment is RNA. And where? On the 3' overhang. On the 3' overhang. Then, coming to DNA repair. There are segments for DNA repair. Then, okay. Before we get to the next, we have mitochondrial DNA and the nuclear DNA. The mitochondrial DNA is a closed circular DNA. The importance, there's very important one. The double-stranded and the linear one is the nuclear DNA. They will always ask in comparison. If they are asking about mitochondria, they will compare it to the nuclear DNA. Then we get our mitochondria only from mother, so it is 100% maternal inherited. There are no introns. Then there is arranged in single chromosome. They are arranged in several chromosomes, the nuclear DNA. You have around 16,000 base pairs for mitochondria, but 3 into 10 to the power 9 base pairs in the nucleus. And you will get the mitochondrial DNA only from maternal inheritance. There is no Mendelian law followed for mitochondrial DNA. Then we have DNA repair. The next is nucleotide excision repair, that is done for pyrimidine dimers. The dimers which are formed by the thymine dimers. This is due to UV radiation. It's written here. It is due to UV radiations or chemical carcinogens. The disorder which it results is number 1, xeroderma pigmentosa. Number 2, write it down, trichothiodystrophy and Cockayne syndrome. These are the disorders trichothiodystrophy. These are the disorders which are seen due to defective nucleotide excision repair. Then there is base excision. This kind of xeroderma pigmentosa picture is seen, and this is extreme form of blistering of skin or hyperpigmentation is seen. Then base excision repair, it is alteration of just one base, cytosine to uracil, or deamination, which is done by APDNA glycosylase. Base excision repair, if this is defective, it is associated with breast cancer, polyposis colon cancer, and please note there is non-polyposis colon cancer which is seen with mismatch repair. So mismatch repair results, if this is defective, it results in a disorder HNPCC, hereditary non-polyposis colon cancer, and the mismatch between it is mismatch between the parental and the daughter strand. Then there is homologous end joining. If this can be due to ionizing radiation, X-ray, it can be due to anti-tumor drugs, which causes double strand breaks, single strand breaks, intra-strand crosslinks. It can result into Bloom syndrome, Warner syndrome, Rathman-Thompson syndrome, breast cancer, and non-homologous end joining. If this is defective due to ionizing radiation, X-rays, it results in SCID. Then the next is tRNA. You just have to know the function of each one of them. I'll start with D loop. The D loop here is also known as DHU, dihydrouridine loop. The dihydrouridine loop is responsible for binding to amino acyl tRNA synthetase. It binds to amino acyl tRNA synthetase. You have to remember the function. There can be a question like match the following for all these loops. Then you have an anticodon loop. The anticodon loop is responsible for binding to the codon. The codons are present on mRNA. The codons are present on mRNA and the anticodon is present on tRNA. And then you have T-Cy-C loop, also known as pseudouridine loop. It is this pseudouridine loop which binds to the ribosomal surface. It binds to ribosomal surface. And then in the end we have an acceptor arm where the amino acid will bind. It binds to the amino acid. It binds to the amino acid. Now, this is one very important concept. Please note this slide. This was this year's INICET question. So let's focus on this. If the two fragments are given, out of the two slides, that is out of the two strands, 5 to 3 or 3 to 5, which one will be responsible for the RNA synthesis? Please note, the above one can also synthesize, the lower one can also synthesize the RNA. The point here is that the newly formed strand should only be in a direction for 5 to 3. So it is not above or lower which decides, no. It is just the direction of the RNA polymerase itself has to be 5 to 3. So your RNA polymerase is always from 5' to 3' direction. That means my lower strand will act as template. So this lower strand acts as template. That means the above strand is anti-template. But when you synthesize the mRNA from the lower strand, my mRNA will read like A, U, G, A, U, C, U, C, G, U, A, A. All I have done is I have done complementary to the lower strand. I have done complementary to the lower strand. So my lower strand is template strand and hence I have taken complementary to the lower strand. If you see this carefully, it looks exactly same as that of the above strand. And hence, the above strand is called the coding strand. The one which is coding is not template. The one which is template is non-coding. So the two are not same. The template is the one which is actually helping in the synthesis of RNA, but it looks exactly same of the above strand. All you have to do is replace T with U. And one additional point, this will always be read from 5 to 3. No matter whatever sequence is given to you, if nothing is mentioned on either side of it, you will read it as 5 to 3. So read this as 5 to 3, right? So your lower strand acts as template. The above strand is coding. They can give you a coding strand and ask for mRNA. They can give you a non-coding or a template strand and ask for mRNA. Any of this is, even reverse is possible. They can give you mRNA and ask for coding and non-coding. But your slide should always read from 5 to 3. Then, post-transcriptional modifications are 3 in number. Number 1, which you can see here, fill the first box, is addition of 7-methyl-guanosine. This is called capping. We are adding capping. Number 2, you are removing the introns. Introns never form the part of the mRNA. They are part of the HRNA, heterogeneous RNA, but they are not part of the messenger RNA. Number 2 is splicing, which is done by spliceosomes, which is done by snRNA, small nuclear RNA, which is same as spliceosome. And number 3 is tailing. Number 3 is tailing. Addition of poly A tail. Addition of poly A tail. These are the 3 most important post-transcriptional modifications. And then the codon is always present on mRNA. The anticodon is always present on tRNA. The 3-letter words are made from always U, A, C or G. There is no T in codon. It is always read from 5' to 3'. The codon system is unambiguous. A codon always codes for a specific amino acid, degenerate, non-overlapping, non-punctuating, universal. With the few exceptions seen in mitochondria, it is universal. The exceptions are the AGA, AGG in the mitochondria codes for arginine. In the nucleus, codes for arginine. In the mitochondria, it is a stop codon. AUA is coding for methionine. In the mitochondria, in the nucleus, it codes for isoleucine. Isoleucine. UGA, it is stop codon in the nucleus, but it codes for tryptophan in the mitochondria. With these few exceptions, the majority is same. The rest of all of them are same. There are 2 types of chromatin modification. We have DNA methylation and histone acetylation. Key points. Where does DNA methylation takes place? At CpG island. Where does histone acetylation takes place? At lysine residues. The role of formation of this DNA methylation is in cancer, gene silencing, genomic imprinting, and in mismatch repair. What is the key point in microRNA? two enzymes. Note what is the location of drosha. It is inside the nucleus. Note what is the location of dicer. It is outside the nucleus. What is drosha doing? It is responsible for removing the cap and the tail. It removes cap and the tail. What is dicer doing? It is removing the loop head. It is removing loop head. Both of these are endonucleases. Drosha is an endonuclease. Dicer is an endonuclease. What drosha is removing? Cap and tail. What dicer is removing is loop head. And finally, we come to our last slide. What is the palindromic sequence? We are having, all you have to do, if you get this kind of a slide, all you have to do is write the complementary to it. If it reads same on both 5' to 3' on both these slides, if it reads same on both these strands, then only it is complementary. You can see for others. Yes, it is complementary. If I write for this, will it read same on both the sides? A, G, C, T, T and A, G? No, it is not complementary. And the last one, I have left it for you. Right, do it. All you have to do is write complementary. If it is reading same on the two strands, it will be complementary. If it does not read 5' to 3' on both these strands, it is not complementary. So do not consider Malayalam to be palindromic sequence. This is where your restriction endonucleases will act. They only act at palindromic sequence. There was one such question this year INICET where they have given you a few sequences and they asked which of them is a palindrome. All you had to do was write complementary to it and see if 5 to 3 matches on both these strands. Thank you all, and I wish you all the best for your upcoming exam. Right, thank you.